Hearts from genetically modified pigs could soon be transplanted into cardiac patients after an organ implanted into a baboon survived for more than 2.5 years. Scientists hope the procedure, known as xenotransplantation, could combat the dire shortage of organs from human donors, which often leads to patients dying before they receive a transplant. Using a technique described as immunomodulation, researchers say they have overcome the natural rejection that occurs when tissue from another species is implanted into a body. Normally the immune system attacks organs and tissue that it does not recognize. But scientists based at the National Heart, Lung and Blood Institute in Maryland used hearts from pigs that had been genetically modified to make the organs more tolerable to baboons. They were then transplanted into 5 baboons, which were given a combination of drugs to alter their immune system so it would not attack the organs. Dr. Muhammad Mohiuddin, a chief of the transplant section of the Cardiothoracic Surgery Research Program at the National Heart, Lung and Blood Institute, said the breakthrough was a big step towards delivering xenotransplantation in humans. He said, "It is very significant because it brings us one step closer to using these organs in humans." Xenotransplants, organ transplants between different species, could potentially save thousands of lives each year that are lost due to a shortage of human organs for transplantation. The researchers, whose work is published in the journal Nature Communications, used pigs that had undergone 3 genetically modifications so their organs appeared less alien to the recipient animal's immune system. In a previous study, researchers had shown that using organs from these pigs alone had allowed them to survive for up to a year in a baboon. But in the new work, the researchers also used a cocktail of antibodies and drugs that allowed them to fine-tune the immune response in the baboons so they did not reject the transplant. 5 of the animals were given the modified pig hearts, which were connected to the baboon's circulatory system alongside their own original heart. While the implanted pig organ did not replace the baboon's heart, they were found to continue beating for up to 945 days with the help of the antibody and drug treatment. On average, the implanted hearts kept beating for 298 days. Writing in the journal, the researchers said they now hope to begin conducting human trials using the technique. They said, "Preventing xenograft rejection is one of the greatest challenges of transplantation medicine. Genetic modifications combined with the treatment regimen tested here consistently prevent humoral rejection and systemic coagulation pathway dysregulation, sustaining long-term xenograft survival beyond 900 days. In our opinion, the regimen appears potentially safe for human application for patients suffering from end-stage organ failure who might be candidates for initial trials of xenotransplantation."